Hello everyone. Welcome to the fourth module of the paper Cell Biology. In this module, we will study about the ultrastructure of the cell membrane and the various subcellular organelles such as the Golgi bodies, endoplasmic reticulum, mitochondria, ribosomes, lysosomes, peroxisomes, chloroplast and nucleus. This module would give us a fairly good idea about the ultrastructure of a cell and the functions of the various organelles present inside the cell. Structure and function of cell membrane. The outer walls of a house or car provide a strong inflexible barrier that protects its human inhabitants from an unpredictable and harsh external world. Similarly, we might expect the outer boundary of a living cell to be constructed of an equally tough and impenetrable barrier because it must also protect its delicate internal contents from a non-living and often inhospitable environment. Yet, cells are separated from the external world by a thin, fragile structure called the plasma membrane, that is only 5 to 10 nanometer wide. Since it is very thin, no hint of the plasma membrane is detected when a section of the cell is examined under a light microscope. It wasn't until the late 1950s that techniques for preparing and staining tissue had progressed to the point where the plasma membrane could be resolved by the electron microscope. Major functions of membranes in the life of a cell. Compartmentalization. Membranes are continuous unbroken sheets and they enclose compartments. The plasma membrane encloses the contents of the entire cell, whereas the nuclear and cytoplasmic membranes enclose diverse intracellular spaces. The various membrane-bound compartments of the cell possess different contents. Membrane compartmentalization allows specialized activities to proceed without external interference and enables cellular activities to be regulated independently of one another. Scaffold for biochemical activities. Membranes enclose compartments but are also a distinct compartment themselves. Because of their construction, membranes provide the cell with an extensive framework or scaffolding within which components can be ordered for effective interaction, providing a selectively permeable barrier. Membranes prevent the unrestricted exchange of molecules from one side to the other. At the same time, membranes provide the means of communication between the compartments they separate. Transporting solutes. The plasma membrane contains the machinery for physically transporting substances from one side of the membrane to the other, often from a region where the solute is present at much higher concentration. The plasma membrane is also able to transport specific ions, thereby establishing ionic gradients across itself. Responding to external signals. The plasma membrane plays a critical role in the response of a cell to external stimuli, a process known as signal transduction. Membranes possess receptors that combine with specific molecules or ligands having a complementary structure. Different types of cells have membranes with different receptors and are therefore capable of recognizing and responding to different ligands in their environment. The interaction of a plasma membrane receptor with an external ligand may cause the membrane to generate a signal that stimulates or inhibits internal activities. Intercellular interaction. The plasma membrane allows cells to recognize and signal one another, to adhere when appropriate and to exchange materials and information. Energy transduction. Membranes are intimately involved in the process by which one type of energy is converted to another type, that is, energy transduction. The most fundamental energy transduction occurs during photosynthesis when energy in sunlight is absorbed by membrane-bound pigments, converted into chemical energy and stored in carbohydrates. Membranes are also involved in the transfer of chemical energy from carbohydrates and fats to ATP. Ultrastructure of cell membrane. Cell membranes are crucial to the life of a cell. The plasma membrane encloses the cell, defines its boundaries and maintains the essential differences between the cytosol and the extracellular environment. Despite their different functions, all biological membranes have a general structure. Each is a very thin film of lipid and protein molecules held together by non-covalent interactions. As you can see from this figure, the lipid molecules are arranged as a continuous bilayer about 5 nanometer thick, and the protein molecules span the lipid bilayer, mediate nearly all the functions of the membrane. Cell membranes are dynamic, fluid structures, and most of their molecules move about in the plane of the membrane. The lipid molecules are arranged as a continuous double layer about 5 nanometer thick. The lipid bilayer provides the basic fluid structure of the membrane and serves as a relatively impermeable barrier to the passage of most water-soluble molecules. Protein molecules that span the lipid bilayer are called transmembrane proteins, mediate nearly all of the other functions of the membrane, transporting specific molecules across it. The lipid bilayer. The first proposal that cellular membranes might contain a lipid bilayer was made in 1925 by two Dutch scientists, E. Gorter and F. Grendel. The lipid bilayer provides the basic structure for all cell membranes, and it is easily seen by electron microscopy. All of the lipid molecules in cell membrane are amphiphilic, that is, they have a hydrophilic or water-loving polar end and a hydrophobic or water-fearing non-polar end. In this diagram, you can see the arrangement of amphipathic lipid molecules arranged to form a lipid bilayer. The polar head groups separate the hydrophobic tails from the aqueous cytosolic and extracellular environments. A phospholipid molecule has a strongly nonpolar and hydrophobic tail region represented by fatty acid chains and a strongly polar or hydrophilic head region represented by the phosphate group. Due to this property, a phospholipid placed in water forms a lipid bilayer in a characteristic manner. Such an arrangement forms a basic component of plasma membranes.